students. You are welcome to School in the Air. My name is Eyitayo Adeoye. I shall be taking you basic science. And the topic today is sanitation. Sanitation. I welcome you once again. I want you to look around where you are seated at home. It's your living room where you are a clean place. I want you to justify it by yourself. Because all what we are going to talk about today centers on how clean your environment is. Don't forget the topic once again, sanitation. At the end of this lesson, you should be able to explain the following terms: sanitation, refuse, and sewage. Sanitation, refuse, and sewage. Now, what is sanitation? What is sanitation? Sanitation is the hygienic means of promoting health through prevention of human contact with the hazard of waste, as well as the treatment and proper disposal of refuse and sewage. The hygienic means of promoting health through prevention of human contact with the hazard of waste, as well as the proper disposal of refuse and sewage. Now, when we are talking about sanitation, we are talking about the removal of all kinds of waste from our homes and our environments. Why do we need to remove all this? Because we said they are poisonous to us, they are hazardous to our environment. Because the human contact with the waste can cause series of problems. Now, it will interest you to know that our God also is really interested in you being clean. The word of the Lord says, "Who shall ascend to the hills of the Lord? He that has clean hands shall obtain mercy from the Lord." In other words, to have good relationship with God and fellow human, you have to be clean. The Lord wants us to be clean. Now, let's go further. What are the tools that we use for sanitation? Tools for sanitation. We have broom, mopping stick, hoe, cutlass, rake. All these tools, we use them to take away, to remove dirt from our environment in order to have clean environment. We use those tools to what? To remove dirt from our environment. As you will see in the picture being displayed, the materials, the tools that we use to take away dirt in our environment. Sanitation. Sanitation is majorly done by disposal of refuse and sewage. Refuse and sewage, proper disposal. Now, what do we mean by refuse? Refuse. What is refuse? Refuse is the combination of dry and solid waste materials. Can you give me an example? Papers, glass, cans. They are solid. These materials I've mentioned are solid and dry. So we group them under the word refuse. Refuse. Now, having known the meaning of refuse, the combination of dry and solid waste. This refuse we are talking about, the processes of refuse disposal. First is collection. Collection. What do we mean by this? Refuse is collected from house to house by waste management agency. You are not to dump your refuse somewhere anyhow. These are collected and for what? For further action. You can ask me, what is the action or what are the actions that we are going to perform after this? From the picture being displayed on the screen, you will see the waste truck where the waste management agency are collecting the waste from house to house. So that is collection. The first point is collection. And the second one is what we call sorting. Sorting. What do we do at this stage? We sort those materials, these waste materials, into different materials. We have plastic, we have papers, cans, and even some materials that can decay. So they sort them out into these groups. But largely, this sorting can be in two groups. Now, the first one is what we call biodegradable refuse. Biodegradable refuse. And the second one is non-biodegradable refuse. Non-biodegradable refuse. Now, let's just go back into what we mean by biodegradable refuse. Biodegradable refuse are refuse materials that can decay. They can decay, which means that they can be broken down by microorganisms that can act on them and break them down. So we say they can decay. Can you give me an example of waste that can decay? I want you to just think about the waste from your house or you have come about that can decay. Oh, don't forget the peels of yam, banana, potatoes, the food remnants. What about your vegetable stocks? All these materials can decay. And because they can decay, they are valuable. You have something you can use them for. They are not to be discarded. They are not to be thrown away like that. They can form compost manure. So when we dispose this, don't forget we are talking about refuse disposal. So when we dispose this, they are being buried to form manure, which can help in agriculture. It can help in agriculture. Now, don't forget what we are talking about, biodegradable refuse. Biodegradable refuse. Now, the second group of refuse is what we call non-biodegradable refuse. Non-biodegradable refuse. These non-biodegradable refuse materials, they cannot decay. They cannot be broken down by microorganisms. So we call them non-biodegradable. These materials are plastics, cans, glasses, nylon. They cannot decay. And because they cannot decay, how do we dispose them? Now, how do we dispose these materials? Because they cannot decay. We cannot just leave them lying fallow or constituting nuisance in our environment. We need to dispose them properly. Now, the first thing we can do to non-biodegradable waste materials is that we can recycle them. And that's what we call recycling. Recycling. I know you have heard about the word recycling. It's the process of converting waste materials into new materials and objects. So we recycle this material. We can use them again. So they are not totally useless. We can recycle them. Example of materials that we can recycle, we have glass, papers, and so on. We can recycle these materials. They are useful. We can recycle them. And you can see the picture on the screen, recycling. The second thing we can do to non-biodegradable waste materials is we can burn them, combustion. Combustion. We can burn this refuse. They are materials that fire can consume them. Combustible materials or combustible refuse materials are burnt in incinerator. They are burnt in where? Incinerator. Why? You can ask. What is incinerator? The picture is on the screen. The picture of an incinerator. Why do we need to burn? It is improper for us to burn in open air. We have to burn in incinerator to avoid air pollution. Air pollution can also deplete our ozone layer. And because of this, we have to burn our refuse or waste in incinerator. In an incinerator, the picture of incinerator being displayed on our screen. Now, the third thing we can do to non-biodegradable waste is that we can use them to fill the land. That is land filling. Land filling. We use this refuse to fill the land in construction. You also see the picture on your screen. Land filling. Land filling. Don't forget, students, we are talking about sanitation. We said that we've talked about refuse disposal. That refuse is the combination of dry and solid materials. And because of that, we group them into two: biodegradable refuse and non-biodegradable refuse. We said that biodegradable refuse, they are the categories of refuse that can decay. The category of refuse that can decay. And I gave you some examples on that. And also that the second one is non-biodegradable refuse. They are refuse or waste that cannot decay. And because they cannot decay, what can we do? We can use them, we can recycle them, we can burn them, and we can use them to fill the land. So, let's go further about sanitation. Sanitation. The second thing we want to talk about is what we call sewage. I told us that sanitation is majorly done by disposal of refuse and sewage. Disposal of refuse and sewage. So, let's go to sewage disposal. Sewage disposal. What is sewage? Sewage is the mixture of human and liquid waste. Mixture of human and liquid waste. But today, in the course of this study, we only focus on human waste. Human waste. Can you tell me the meaning of sewage? We said sewage is the mixture of human and liquid waste. Now, when we say human and liquid waste, and we have said that we are focusing only on human waste today, what are the methods of disposing human waste? Methods of disposing human waste. Human waste is disposed of in the following ways. 1. Pit latrine. Pit latrine. We have pit latrine. It is in Nigeria here we have a method we call pit latrine. As you can see the picture being displayed on the screen now. Pit latrine. And another thing that we have in Nigeria here, we also use pail or bucket latrine. Pail or bucket latrine. From the word, you could get the means of disposing this, using a pail or bucket to dispose human waste. The third method is what we call open or bush defecation. Open or bush defecation. Some of us at home, students, you may be feeling somehow that what are this method all about? Open or bush defecation. It's a method by which we dispose our sewage, human sewage. And the last point I will talk about under sewage disposal, which is very comforting, is water closet system. Water closet system. Oh, what a relief. Water closet system is commonly used these days. Now, students, don't forget we are talking about sanitation. The proper disposal of refuse and sewage. You can just be wondering, why do we need to properly dispose sewage and then refuse? It is of great importance. And I want you to follow me into the importance of sanitation. Importance of sanitation. Why do we need to have clean environment? Why do I need to live in a clean place? What are the benefits I am going to get in living in a good or clean environment? Point number one, prevention of diseases. What do I call it? Prevention of diseases. Students, even I want you to know at this period of COVID-19 pandemic, we are told to clean our hands, clean our environment, maintain personal hygiene. Why? To prevent us from contracting this disease. And that is also very related to what we are talking about, sanitation. When you have clean environment, it tends to prevent diseases. So we need to have what? Maintain clean environment. Point number two is that it promotes good health. It promotes good health. Yes, you should know that waste do breed different disease vectors. But once waste is removed from our environment, we tend to have good health, because all these disease vectors, they carry disease from one place to another. And if we continue to breed them by putting our waste, dropping them anyhow, it tends to affect our health. And then our health or the health of an individual is being challenged. Therefore, we have to do away with all these waste materials from our environment in order not to breed disease vectors. Now, we've talked about two importance: prevention of disease and promoting good health. Now, let's go to another point: good farming. You can wonder and say, what is the correlation between sanitation and farming? This will take us back a little when we're talking about refuse. Have you forgotten? I said that we have biodegradable refuse and non-biodegradable refuse. The biodegradable refuse, I said, they are examples. I gave example of the peels of banana, potatoes, the food remnants. These materials, they can decay, and because of that, they can form compost manure, which we bring good produce from our farming. So biodegradable waste help to boost our agricultural produce, since it can be used to form compost manure. So good farming. It is even better for us to use natural materials to produce manure than our artificial fertilizers. Now, another point I would like to talk about under the importance of sanitation is prevention of pollution. Prevention of pollution. Sanitation, when our environment is clean, you should know you will have good smell all about. So, refuse and dirt generate unpleasant smell, causing pollution of air. Therefore, when we have good and clean environment, we always have very good air to breathe in. So, under the importance of sanitation, we have talked about 4 points. I want you to also think about other importance of sanitation. Students at home, I want us to quickly go over what you have learned today. I believe by now you are able to explain sanitation. That sanitation is the what? The hygienic means of promoting health. Hygienic means of what? Promoting health through prevention of human contact with the hazard of waste, as well as the treatment and proper disposal of refuse and sewage. And you are able to learn that refuse is what? The combination of solid and dry waste. That refuse can be grouped into two: biodegradable and non-biodegradable refuse. The biodegradable refuse are refuse materials that can decay. They help in what? To boost our agricultural produce because they can form compost manure. We have talked about sewage disposal. That sewage is the mixture of human and liquid waste. And that we have some methods of disposing sewage. We talked about water closet system, which is the best system to adopt to avoid air pollution in our environment. We talked about pail or bucket system. We talked about open defecation. All these methods to dispose our sewage. And I want you to know, students at home, that you should maintain personal hygiene and clean environment. It is very, very important to what? To have all the benefits we have discussed, the importance of sanitation. And don't forget that God is interested in you being clean. Now, there's a wise saying that cleanliness is next to godliness. Till I come your way next time. Bye.
welcome to Christ the Redeemer School Management virtual class. The subject we are considering today is chemistry. My name is Olubunmi Thompson Akinawunu, and the topic we are going to consider is qualitative analysis. What did I call it? Qualitative analysis. I believe you can repeat that after me. Qualitative analysis. Don't forget, qualitative analysis is an aspect of chemistry practical. The other aspect is quantitative analysis. Qualitative analysis. So we have to define qualitative analysis first. Qualitative analysis involves identification of anions and cations that are present in ionic compounds. It also involves identification of functional groups that are present in organic compounds. Let me repeat that again. Qualitative analysis involves identification of anions, which are negative ions, and cations, which are positive ions, that are present in ionic compounds. It also involves identification of functional groups that are present in organic compounds. Because qualitative analysis is very, very broad, in fact, it is not possible to conclude it on a single day like this. So we are going to look at a subtopic, and that subtopic is what I call identification of cations. I think you are going to say that after me. Identification of cations. Identification of cations. Good. Now, what are cations? Cations are positive ions. Cations are positive ions. Don't forget, there is also anions. Anions are negative ions, but today we are considering cations. But before we proceed further on cations, do you know that generally everything in nature is of two sides? There are two sides to a coin. We call it head and tail. If you have a bar magnet, you have the North Pole and you have the South Pole. If you have a battery, some of you have seen battery, maybe the car battery, for example, you have the positive terminal and you have the negative terminal as well. Everything in nature almost everything is having two sides. Let me give you another example. Do you know that you have good and bad? Do you know that you have tall and short? Like that, like that, like that. Maybe that is the reason why God created heaven and earth. He equally created day and night, and he created light and darkness. He created us either as a male or as a female. I'm happy to tell you that we have something like that too in chemistry. We have positive ions and we have negative ions. When these two ions combine together, a compound is formed. So don't get confused with ion like this, I-O-N, and ion like this. The two of them have the same pronunciation. You pronounce them the same way, but they are not the same thing. If you are a good chemistry student, you will know that this ion is what? Think of it. Is what? This is an element. It's an element, and the symbol is Fe. But this ion, I-O-N, is a charged particle. When we say a charged particle, we are saying that a substance that is carrying either a positive charge, which is cation, or a negative charge, which is anion. It is possible for an element like this, which is ion here, to even become ion like this if it carries a charge. For example now, if you have something like this, Fe2+. This Fe is ion and is carrying a charge, so we call it ion-2 ion. Now, let's look at examples of cations. By the time you write practical exam in SS3, you must know how to test for some cations. These cations are calcium ion, we have copper 2 ion, we have iron 3 ion. All of them, you will see them on the screen. We have aluminum ion, we have iron 3 ion, we have lead 2 ion, we have ammonium ion, and we have copper 2 ion. And you must know how to test for all these ions. Let me give you an example of these ions again. We have calcium ion, we have iron 3 ion, we have copper 2 ion, we have ammonium ion, we have lead 2 ion, we have zinc 2 ion, and we have aluminum ion. You will see them on the screen. So you must know how to test for these cations. But today, we are not going to test for all the cations because of time. What are the materials or the apparatus that we need to carry out qualitative analysis? And for example, if you want to test for cations like this, you have some of them with me here. By the grace of God, I'm going to show you some of these materials. This is known as a test tube. A test tube is made of glass. I believe you can see it very well. A test tube. The bigger one is known as a boiling tube. So you have a test tube, you have a boiling tube. If you compare the two, you see that this is bigger than this. So this is a boiling tube. We use the two of them for almost the same thing, except that when boiling is required, it is preferred to use boiling tube. So I've shown you two materials now. We have test tube and boiling tube. Then apart from test tube and boiling tube, there is something here that we call spatula. This is spatula. Look at it very well. It looks like a spoon, but this is not a spoon. You are not allowed to use this to eat. We use spatula to scoop chemicals, particularly dry chemicals. You are not allowed to take chemical with your bare hands, so you have to use spatula. Apart from spatula, we have hand gloves. I believe you can see that I'm wearing hand gloves. Is it? Hand gloves. I'm not wearing this because of COVID-19 pandemic. I'm wearing it because I'm about to perform an experiment. Anytime you want to perform an experiment in the lab, it is expected of you to wear hand gloves. Apart from hand gloves, we also have this that we call test tube rack. Test tube rack. Let me try to lift it up for some of us to be able to see it very well. This is a test tube rack. As the name implies, test tube rack is for holding test tubes, because if you put test tube on a table directly, it may fall. And because most materials, apparatus in the laboratory, they are made of glass. So if it falls, it will get broken. So you have to put them in test tube rack. Another material which is very, very important is something that looks like apron that I'm wearing. We call it laboratory coat. So when you are performing an experiment in the laboratory, you must wear your lab coat. And there is also this also. This is known as wash bottle. Wash bottle. Wash bottle is for drop-wise addition of water in the course of a chemical reaction. Wash bottle. All these apparatus that I've mentioned earlier. And now we want to look at the reagents that are needed for qualitative analysis. What are the reagents that are needed for qualitative analysis? Two chemical reagents are needed. And these two chemical reagents are sodium hydroxide solution. This is sodium hydroxide. The formula is written on it, NaOH, AQ. When you see AQ, it's telling you that it's in solution form. AQ actually means aqueous solution. Sodium hydroxide is a chemical substance. And also, you have ammonia solution. The formula for ammonia is NH3. We equally call it aqueous ammonia, NH3AQ. So these are the two reagents that are needed when you are carrying out tests for cations, or you want to identify cations in a compound. All right. The next thing is the procedures. How do we carry it out? What I'm going to show to you today is not magic. That is why chemistry is very interesting, because you'll be able to see it yourself. So the procedures, just about 4 procedures, and you are good to go. What you need to do is to prepare the solution of the sample you are testing for. You prepare the solution of the sample. After that, you divide the solution you have prepared into 2 test tubes. You divide them into 2 portions, each portion in a separate test tube. You will see the procedure on the screen. Then, after that, the first portion, you add sodium hydroxide solution in drop, and after adding it in drops, you add it in XX. As you are doing it, you try to observe what is happening. The next thing is to take the second portion. To the second portion now, you add ammonia solution in drops and in excess. And you also try to observe what is happening. After your observation, you are to conclude. And the conclusion in this case is referred to as inference. Now, because this is not a theoretical aspect of chemistry, it is actually a practical aspect. And that is why I want you to see this thing. This is a compound. In fact, it's an ionic compound. That is to tell you that it has positive ion and it has negative ion. I'm going to pour it out for you to see very well, but I believe you can see the name. The name is cupric sulfate. That is the old name, cupric sulfate. But the IUPAC name, which you must be familiar with, is copper 2 tetraoxosulfate 6. Will you say that after me? Copper 2 tetraoxosulfate 6. I believe you know how to write that formula. That is another topic for another time. But let me show you what you have here. Let me pour it out for you to see. If you look at it very well, you see that the color is blue in color. Most copper salt, not all, have blue color. So when you look at this, don't think it is detergent. This is a salt. And when we are talking about salt, don't think it is only sodium chloride, NaCl, that we eat at home. It is true that sodium chloride is a salt, but that one is a table salt. Other salt that you have in the laboratory are industrial salt. You don't eat industrial salt. Some of these salts are carcinogenic, some are toxic, and some are corrosive. So that is the reason why we have to wear hand gloves and use spatula to take the quantity that we need in the course of an experiment. So this is copper 2 tetrasulfate 6. We are going to follow that 4 procedures now and see how we can identify copper 2 ion in it. Don't forget the procedures. I said, prepare a solution of the sample that you want to test for. And to prepare a solution, what do you need? You just need to pour small quantity of water in a test tube or in a boiling tube. Take your spatula, take small quantity of the salt, add it, and shake so that it will form a homogeneous solution. Whenever you want to test for cation and you want to use sodium hydroxide and ammonia solution, the salt that you are using must be a soluble salt. So this is a soluble salt now. What is a soluble salt? A soluble salt is a salt that dissolves in water. So if you see this, how do you know that a salt has dissolved in water? There won't be any particle left after shaking. So copper 2 tetrasulfate 6 is a soluble salt. So the first procedure is to prepare the solution of the sample. I believe you can see it very well. Now, the next thing is that we divide that solution into two. We divide it into two. Let me take another test tube. Divide it into two. Okay, we have done that. The third thing to do is to add sodium hydroxide solution in drops at first, and then you add it in excess later. I've shown you this before. What is this again? This is sodium hydroxide. Most chemical reagents like this, they look like water. That is why when you are in the lab, don't think it is water and don't drink it. In fact, you are not even supposed to eat in the lab in the first instance. So this is not water now. That is why reagents in the chemistry lab are labeled. This is sodium hydroxide. So we add it to the first portion of what we are having. We add it in drops first. I've added about 2 or 3 drops. If you look at it, the color is blue. Blue gelatinous precipitate is formed. When you say a precipitate, it means an insoluble substance has been formed. And it looks like gel. That's why we say it is gelatinous. Now let's add excess to it. The reason for adding it in excess is to see whether this blue gelatinous precipitate will still be there or whether it's going to dissolve. So let's add it in excess. The blue gelatinous precipitate is still there. It's not dissolving. So if you want to write this now, well, our observation in this case is going to be blue gelatinous precipitate in drops, and in excess, the blue gelatinous precipitate is insoluble. We take the second portion and we add ammonia solution this time around. We add ammonia solution. And whenever you are using chemicals, you have to be very careful. Ammonia smells like urine. Will you imagine somebody that has urinated and leave it for 10 days? That is the way it smells. Maybe when you get to your school, ask your chemistry teacher to show you ammonia. But it must be a dilute ammonia because the concentrated ammonia is corrosive. So I'm going to add ammonia now in drop at first. Drop. Good. Let me see. You have to be careful when you are adding ammonia because if you add excess, something is going to happen. So you have to add it in drops. So in drops now, let me add maybe just another drop for you to be able to see it well. Okay. So I've added ammonia. You see? Pale blue gelatinous precipitate is formed. Blue gelatinous precipitate is formed. Let's add excess to it. The excess is to see whether the gelatinous precipitate we are having will dissolve or whether it will still be there. What do you observe here? Why I said that one was pale blue, can you compare this? The first one looked like this when you added it in drop. But this time around now, what color is this? I believe you agree with me that this is a deep blue color. So when you add ammonia in drops to any solution that contains copper 2 salt, you are going to have pale blue gelatinous precipitate. But by the time you add it now in excess, you discover that that pale blue gelatinous precipitate will dissolve. You will not see the gelatinous there again. It will dissolve to form a deep blue solution. And this is where you have to be careful when you are writing your practical exam. Many students cannot differentiate between a precipitate and a solution. A solution is clear, but a precipitate, you see something like particles telling you that that substance is insoluble. So the correct way of writing the observation in this case is when sodium hydroxide is added in drops, a pale blue gelatinous precipitate is formed, and when it is now added in excess, a deep blue solution is formed. The precipitate dissolves to give you a deep blue solution. The precipitate dissolves to give you a deep blue solution. When you are carrying out this experiment in the lab, or maybe in an examination condition, the name of the salt will not be given to you. That is why this salt is labeled as X. It's labeled as X. So in the first experiment we performed, we are going to refer to this salt as X, as well as in the second experiment. So after you have performed this experiment now, how do you present it? What do you do next? The next thing is reporting of the experiment. Reporting of the experiments. Now, there is a standard for reporting these experiments. There's a standard. And the standard is what we call a 3-column table. You have the test, you have the observation, and you have the inference. You see it on the screen. All what we have done under test, you have it there. Observation is there as well, and the inference is equally there. So far, so good. We have come to the end of the class for today. But before I will give you the assignment that I want you to work on, do you know that our Almighty God is the greatest chemist ever? Even when we have not been created, in his infinite wisdom, he combined hydrogen ion, a positive ion, with hydroxide ion, a negative ion, to form water, which is indispensable. God is very creative, and he wants you, as a chemistry student, to be creative too. So, I'm going to give you an assignment, as I've said, and the assignment is already displayed on the board. Describe how you identify zinc ion using sodium hydroxide solution and ammonia solution. Describe how you identify zinc ion, that is zinc 2 ion, using sodium hydroxide solution and ammonia solution. Until I come your way next time, bye and God bless you.